inheritance can be in two ways. Perspective again. Functionally and structurally. When we talk about inheritance functionally, what do we speak about? We speak about gene inheritance. Genes. So Mendelian inheritance talks about the functional inheritance. There are characters. Characters are represented by a dominant factor and a recessive factor. The presence of a dominant factor will always bring about an expression, irrespective of the other allele being dominant or recessive. Then Mendel talks about independent assortment. Mendel talks about all these principles. The basis of Mendel's understanding of inheritance is that the factors are particles. Factors are particles. That is, each gene, when we are talking about factor from the Mendelian perspective, Mendelian point of view, we are talking about gene in our current understanding. So what Mendel called as factor then is the gene that we refer to today. So if I use gene and factor interchangeably, you should understand it is the same. So Mendel gave all those theories, law of segregation, law of independent assortment, all of that, based on the understanding that there are as many particles in pairs as there are characters that are expressed. So while segregation happens, while segregation of these factors happen during sexual reproduction, during gamete formation, the two units of the particle, the two units of the factor will move randomly into different gametes. Meaning each gamete will get only one unit of every factor. So according to him, how many units are there in every factor? In every gene? 2 units. Each of the individual units he referred to as alleles. Both the alleles put together is the factor. Factor is a reference to the unit of inheritance, is a reference to the part of inheritance that brings about expression. Factor is the reference to the character. So factor for height will control height. Allele will control expression. Allele will control the range of expression. Law of independent assortment all worked out in Mendel's experiments. Whatever experiments he did, he was lucky in a way and unlucky in many ways. Lucky in a way because his experiments gave him consistent results by virtue of the plant that he selected being pea plant, limited number of characteristics, the characters he chose to combine and experiment with multifactorial inheritance, those characters were not closely linked characters, which gave him results to justify his hypothesis. There are linked characters also in the pea plant's genotype. If he had selected those characters for his experiments, his results would have been inconsistent. But it's good. Good that he did experiments on so factors, on such factors, which gave him consistent results, which gave us the basis of understanding genetics. Keep that aside. Today, with our understanding, what do we know? Is it factor-based inheritance or chromosome-based inheritance? What gets segregated during gamete formation are not factors but chromosomes. Every chromosome has multiple locus. Locus is the location of a particular gene on a chromosome. Okay? So today the factors are not particles, the factors are segments on the chromosome, part of the chromosome. There are long chromosomes. There are chromosomes are what are chromosomes? Stretch of DNA. DNA coiled around histone proteins. DNA is coiled around histone proteins. So when you condense it during the application, you see it as chromosomes. But if we go even further down, gene or factor is a segment of DNA. That's all. Is a segment of DNA. If you want to bring it down to chemical representation of the gene, it would be a particular sequence of nucleotides. That is what we can say. That is the language of the gene. So a series of nucleotides in a particular sequence is a particular gene. Different genes will have different sequences of nucleotides. It is like nucleotides are the alphabets on the DNA. Genes are the sentences or words, you can say. Genes are the words. With the same 26 alphabets, we have millions of literature being created, different combinations. Similarly, with four nucleotides in combination and in varying lengths of nucleotide traces, you have a number of genes that are created. That's all. So what is a gene now? Inheritance is not of particles. This is not being segregated into two alleles and this goes out. It is chromosome. Let us say this is the chromosome. This is one chromosome that we are writing. One stretch of DNA. See, DNA is the right way, but we will talk about it in terms of chromosomes, condensed. So the segment becomes smaller. A particular segment of the chromosome which represents a particular gene is called locus. Locus is the location of the gene on the chromosome. It doesn't mean there is a chromosome and there is gene on the chromosome. That segment of the chromosome itself is the gene. Alright? So when we say locus, the definition is the location of the gene on the chromosome. The right way to look at it is locus is that segment of the chromosome which represents a particular gene. The locus of a particular gene is a segment of the chromosome which represents that gene. It is not the location of the gene on the chromosome. Understand? Now we know this. During DNA replication, during meiosis, during the interphase of meiosis, something special happens. What happens? Crossing over. Can you recall the stages of interphase of meiosis? No, interphase or prophase of meiosis. Can you tell me the stages of the prophase of meiosis? Huh? Correct, I heard it all. Be louder, if you are confident about it, be louder.
Leptotene, zygotene, pachytene, diplotin and diakinesis.
Leptotene, zygotene, pachytene, diplotin and diakinesis. Have you watched a movie called Dilwale Dulhaniya Le Jayenge? That is prophase of meiosis, climax scene. What happens in the climax scene? Train is going. Shah Rukh Khan is on the train at the door. Kajol comes running towards him. Correct? That is leptotene. The homologous chromosomes come closer to each other. They hold hands. That is, what is that happening? That is the zygotene. There is a segment where they come in, where the two homologous chromosomes come in contact with each other. Zygotene. Diplotin is exchange. Recombination happens. Exchange of material. So then diplotin, zygotene, pachytene is exchange of material, recombination. Diplotin is forming that one, when you are pulling them away, you have that one, what is that called? Chiasma. That one chiasma, pulling away that from holding hands to just having contact with the fingertips and then diakinesis separate. You change the climax of DDLJ, Dilwale Dulhaniya Le Jayenge, and here Shah Rukh Khan and Kajol are not lovers. Shah Rukh Khan is a drug dealer. Kajol is the drug supplier. Shah Rukh Khan has a briefcase of money Kajol has a briefcase of drugs. Now he is moving. Her father, Amrish Puri, is the police. He is holding the drug supplier. Now she has to somehow deliver the drugs to the dealer. She is negotiating. मुझे छोड़ दो, मैं तुझे हिस्सा दे दूँगी. I'll give you a part of the money. I have to supply this drug. Without this drug, so many people will die because they are already addicted to it. Do good to humanity. Where is your humanity? Then suddenly Amrish Puri starts thinking, now I got a mask of humanity, but in reality I want money. So I will let her go in the name of humanity, but I will actually collect money when she gets the money. But he looks at it, train is already going. Oh my God, my money is going away. Ja, jile. When she runs, F15, the homologous chromosomes come closer to each other. Contact, zygote, exchange of briefcase, match it in. Then that final goodbye, the separation of the hands, diplotin, then they get separated, diakinesis. That's all. So, chromosomal segments exchange. This is one chromosome, this is one homologous chromosome, this is one homologous chromosome. This is a pair of homologous chromosomes. It is here somewhere in the initial part of prophase. They have condensed, but they have to condense. They have to come closer to each other now if they have to if they have to bring about recombination. Now they come closer to each other. The protein. There is a contact is established. Zygote. Then the segments get exchanged. Patched in. Then they are separating, but there is one small point of contact here. Diplotin, they are fully separated, diakinesis. Now tell me, there is one recombination loop here. Can there be one more immediately next to it? Recombination, can it happen in the immediate adjacent segments? You are forming the loop. So physically, the chromosomes have to form these loops of contact. The chromosomes have to form the loops of contact. The segments that are very close to each other will not be able to form separate loops. As they get farther away, the chances of establishing this recombination loops is higher in the segments that are farther from each other. There can be one here, there can be one more here. There can be one more little further. Alright? Linkage is the proximity of genes to each other. How close the genes are to each other. Very closely located genes will always go together. If exchange happens, the entire segment gets exchanged. Both of them will have exchange, recombination. Otherwise, both of them will not exchange. So linkage does not allow recombination in the true Mendelian sense. Are you understanding? Let's say Mendel did experiments with height, tall plants with round seeds, dwarf plants with wrinkled seeds. Tall and round are dominant characters. Dwarf and wrinkled are recessive characters. All these are possible. All these combinations are possible. Tall, dwarf, round, wrinkled. Now you can have tall, wrinkled, tall, round, dwarf, wrinkled. All these four are possible in phenotypes. All these four phenotypes are possible according to Mendel's experiments. It was possible because the gene for tallness or height is here, suppose it is here. The gene for shape of the seed or the texture of the seed is somewhere far away from this point. That is why this also could bring about recombination. This segment also underwent recombination. This segment also underwent recombination. So because of that, the combinations, all the four possibilities could happen. But if this segment, tall is, the height is here, shape is here, and another segment, corresponding segment It is tall, round. Now can recombination happen here as well as here? It can still happen, but the probability of recombination is lesser. The probability of all the phenotypes being possible is lesser. Correct? One of the gamete will always be this, one of the gamete will always be this. Whatever combinations happen, if the other gamete also is the same, recessive for both height and shape or texture of the seed, you will have dwarf plants with wrinkled seeds. This is also possible. But you don't have this combination gametes. This combination gamete is not possible because this recombination is not happening because of the proximity of the genes to each other, because they are too close to each other. Anybody has tape? Tape. R tape. Or some long weight or something. We have this problem here. Okay. Consider this to be a pair of homologous chromosomes. Alright? See, one recombination happened here already. Right? Now let us say this is the length of the segment. Immediately here is another gene adjacent to this point. Is it easy to make this spontaneously, or is it easy to make this? This can happen easily, spontaneously, but this another crossing over happening at the immediate location, at the immediately adjacent point, requires effort. The probability, it is not absolute, it is not like it can never happen, or it is not like it will always happen. The probability of two crossover loops forming adjacent to each other is lower compared to the probability of crossover loops forming at a distance from each other. Yes or no? So this is, now if you say there are genes here, there is gene A, B, C, D, E, F, goes on like that. Now let us say there is crossing over happening in the gene segment A. There is crossing over happening in the gene segment, let's say G. What is the strength of linkage between A and G? The gene A and gene G are not strongly linked. They are weakly linked. They are still linked because they are all on the same chromosome. By virtue of being on the same chromosome, there is linkage. There is linkage. But the strength of linkage depends on how close they are to each other on the chromosome. Now gene A and gene B, they are strongly linked to each other. If the linkage strength, if the strength of linkage is more, the likelihood of the two genes going as a unit into the gametes is higher. The separation of those two alleles will be different, will be difficult. But if the linkage is weak, recombination chances are high. Crossing over is highly probable. You understand? So this is the context of linkage and recombination. Recombination can happen when the two segments of the homologous chromosomes, corresponding segments of the homologous chromosomes, cross over. What if the non-corresponding segments of the homologous chromosomes cross over and the exchange of genetic material is happening, segment exchange happens, but the gene sequences are disturbed? If the corresponding segments of the homologous chromosomes cross over and the gene is produced and the gene exchange happens, the allele will become altered. The allele gets altered, but the gene still remains the same. What if non-corresponding segments exchange material? Is that impossible? May not be impossible, might happen. If it happens, the gamete will become unviable. The cell that is produced will become unviable. It will not have the strength to carry forward formation of the new organism. Reproduction will not progress. That is why many times the progression of fertilization might happen, but the embryo may not sustain, embryo might not survive, it will just be removed. There are natural mechanisms to ensure that viable embryos move forward in development. Viable gametes will go further to fertilize. If it doesn't happen, that's all. Alright? So we will read it now. We will read from the book and see what else needs to be corrected. Chromosomal theory of inheritance is that only. Mendelian theory of inheritance and chromosomal theory of inheritance. You have two theories. It is just two ways of looking at inheritance. In Mendelian theory or in the Mendelian way of looking at inheritance, we look at inheritance in the form of factors. Factors are looked at as particles. So exchange happens, combinations happen, so variations happen. But in the current understanding, we look at it as chromosomal exchange, chromosomal combination and recombination. So the variations are possible only to the extent of what chromosomes can allow. So you cannot have, still you have a large number of variations. Yes. Not possible for having all possible combinations in variations. Okay, so because of chromosomal pattern of inheritance, the inheritance predictions are easier compared to if it was factorial. P plant 7 characters, 7 chromosomes, 7 genes. Human beings already 20,000 genes are identified. Imagine all possible combinations allowed. We don't know what would have happened in this. Okay, we will just read linkage and recombination. We will not do the entire chapter. Like I have told, we will go topic by topic. We will deal with this. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by Mendel in bees. For example, Morgan hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other and the F2 ratio deviated very significantly from the 9:3:3:1 ratio expected when the two genes are independent. See, a dihybrid cross gives you 9:3:3:1 phenotypic ratio because in both the parental organisms you have all 4 possibilities of gamete formation. If you look at it if you look at the same in this. See, when writing the alleles for any particular factor, you will write the dominant allele first, recessive allele next. Complete the alleles of one factor and then write the factor alleles of next factor. Convention. Okay. So we are crossing a tall plant with round seeds with a dwarf plant with wrinkled seeds. Gametes in all the gametes will have only this possibility because there is no other combination possible. Both the alleles are homozygous. Here also it is all homozygous alleles. So this is the gametes. First generation will be, all of them will be tall with round seeds, but all of them are heterozygous. So F1 parents are both heterozygous for both the characters expressing the dominant character. Now F1 gametes. Take the first allele, combine it with the next allele. You have to take one allele of both. The same first allele with the second allele of the second factor. First allele of the second one, second allele of the first factor, first allele of the second factor, second allele. Same with that also. Same with that also. And when we write a Punnett square, I am running through this quickly because I assume that all of you understand this. When you write the Punnett square, you will write one parent here, gametes of one parent here, gametes of another parent here. Some people have the habit of writing it in a rhombus shape. I prefer a square. Simple, straightforward shapes for now. Convention. Entire thing, if you look at this Punnett square, this entire diagonal will have the same phenotype, genotype. Heterozygous for all of this. Now this one and this one similar. This will be homozygous dominant for both. This will be homozygous recessive. Then this one and here. Anyways, we'll write it first. Alright. Write this Punnett square all of it. Write this Punnett square. I want you to observe a certain pattern. Write it and then I'll tell you the pattern. All of you quickly read this. Chief Minister landing. Speaker. Mark the mirror image blocks. Consider this to be the diagonal and we fold it. This is one. This will also be one. Mirror images. Yes. Then these two. Two. Where will this go when you fold it?
So the second one is capital T.
This one, this no? Huh? Capital T, correct. Oh, I'm getting lost in the geometry of this. Tell me, this is one, this will overlap. this will overlap. You fold it. When you fold it, when you fold this, this and this will overlap. Hmm? 3. This one, huh? 3. So 1, 3, 2, 1, 3, 2. This is the diagonal. This is diagonal. This is the diagonal. 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 Now this will overlap with this, correct? 4, 4, 5, 5. This is 6, 6. Observe the overlapping segments. You see 1, 1. Homozygous for both, homozygous for both. Homozygous dominant, homozygous recessive. So if you look at the ratio for those numbers, this is 1. Start with that. This 2. This is dominant on 1. The genotypic. Both dominant. Both alleles dominant homozygous. The first gene dominant homozygous. The second gene dominant heterozygous. How many are there? The first gene dominant homozygous. Heterozygous. 1 is to 2 is to, oh, forget it. I want you to see a pattern, but I am not able to articulate the pattern. Okay, we'll skip that for now. We'll skip that for now. Now, we'll get back to linkage. We get back to linkage, this fold it. Okay. Those logics are not somewhere written down, so I have to get it back something.
Genotypes are just opposite 1 and 1, 2 and 2.
And it's leave it. Leave it. Okay. So in linkage, if this is always if one, let's say if this is going together. Let's say this is the pair of homologous chromosomes. T and R are together, or let's say T and small r are together. Small t and capital R are together. This is the pair. Let us say this is the pair. Now, you can only have T and small r as one unit and small t and capital R as one unit. You cannot have capital T and capital R, small t and small r as combinations when they are linked.
If you take diagonal this way, then you can extend this.
Diagonal?
But instead of this, you can take this.
This diagonal?
Then it overlaps.
Correct. It is the same, no? This one. Correct. You fold it along this, not along this. You are right? See here. Okay, you see that, no? It is not waste time on that. If you look at this as the diagonal, along that, this is the same, this is the same, the entire thing is the same, these two are the same, these two, this one. Yes. That gives you the genotypic ratio when you fold it. Okay? It folds along the diagonal. It's a mirror image of each other, not along this diagonal, along this diagonal. Yes. Alright. Completely. So, Morgan observed that the two genes did not segregate independently of each other and the F2 ratio deviated very significantly from the 9:3:3:1 ratio, which is expected when the genes are independent. Morgan and his group knew that the genes were located on the X chromosome. He was experimenting on sex-linked inheritance in Drosophila and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. What is the proof of recombination? What is the proof of recombination and independent assortment? Deviant expression of the gene or the expression of characters which are not exactly like either of the parents. That is the proof of recombination that we see in the supplementary gene experiments. the comb of the chicken. Pea comb and rose comb are the parental types. But in recombinants you get walnut comb and straight comb also. Do you remember this? Supplementary gene combinations? No? Walnut comb and straight comb also are combinations. See, what you know what comb is? Comb. You know what complementary genes and supplementary genes are? Complementary genes and supplementary genes. Complementary genes are those separate genes which, when independently expressing as a homozygous condition, will have their independent expression, but when both of them come together, both of them will give rise to the presence of both will give rise to a new characters, separate character. Supplementary genes are two genes expressing separate characters in the presence of each other. In the presence of each other, there is a new character expressed even when the one is also separately expressed. Okay? It's like two of you are there, both of you don't like the same teacher. Both of you don't like the same teacher. When you are independent, you will not do anything. When your friend is independent, your friend will not do anything. But when both of you come together, you will beat the teacher. Complementary gene. Complementary gene. That is, you have a separate expression, your friend has a separate expression. When both of you are present, you have a different expression, common expression. Supplementary gene is, you are independently beating one teacher, your friend is independently beating one teacher. When both of you come together, you start beating up the third teacher. That is supplementary gene. You will see the examples if it comes here. Or I'll show you, I'll tell you the example. Okay. Do you have complementary gene and supplementary gene? You should be having. It's not doing this. Morgan attributed the expression of more parental characteristics to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combinations. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, which shows very low recombination, while others were loosely linked, showing high recombination. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. Student Alfred Sturtevant used the frequency of recombination between gene pairs of the same chromosome as a measure of the distance between genes and mapped their positions on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of the Human Genome Sequencing Project. See, linkage you understood, the proximity of the genes on the chromosome. What is this association of understanding of linkage to the Human Genome Project? How will the understanding of linkage help you in sequencing the human genome? How can we sequence the human genome by the understanding of linkage? When we mapped the human genome was studied in fragments. You did not take the entire length of the chromosome, entire length of the DNA and studied it from one end to the other end. You made fragments of it and then started studying the chromosomes, started studying the DNA sequences. Corresponding sequences, the sequences which are in continuity with each other have to be mapped. Once you make a list of polynucleotides present in every fragment that you study and you run it through the systems, when you run the sequences through software-based analysis, The sequences in continuity are identified by the understanding of the linked gene sequences. You understand? So there is a fragment here. You cut a particular gene here, you get a particular chromosome here. This segment you have mapped. Another segment might have been cut here, that is mapped. The overlapping sequences have to be identified, and then you start reconstructing the entire gene sequences. Understand? So, linkage and gene maps were used for the sequencing of human genome in the Human Genome Project. See, earlier questions used to come in terms of linkage percentages, calculate the strength of linkage, calculate the percentage of linkage. Have you seen those questions? In the earlier question papers you might have found, but it has stopped coming anymore because the calculation of the strength of linkage is not easy. Alright. Okay. Linkage is in contradiction to a few principles of Mendel. What Mendelian principles are contradicted by our understanding of linkage? Certain things that Mendel proposed have been contradicted because we know linkage now. What are those things which are contradicted? What is not consistent with the understanding of linkage vis-a-vis Mendelian principles of inheritance?
Linkage deviates from Mendel's law of independent assortment.
Linkage deviates from Mendel's law of independent assortment. So the prediction of inheritance or the phenotypic expression also is not consistent with what Mendel has proposed. So what Mendel has proposed stayed consistent with his experiments because of the weak linkage that he had, that was there between the characteristics that he studied. But in reality, we don't see the gene expression in consistency with the Mendel's principles of inheritance. There will be deviations from that. Alright. Right. Any other topic from this inheritance? Extension of this? Next topic.
Explain this sickle cell anemia.
What?
Sickle cell anemia.
Sickle cell anemia. Sickle cell anemia is Mendelian disorder or chromosomal disorder? Sickle cell anemia is a Mendelian disorder. What linked? X-linked or autosomal? Autosomal recessive trait. What happens in sickle cell anemia? Is it recessive inheritance or dominant inheritance? Sickle cell anemia. Recessive. Fortunately for human beings, many of the genetic disorders are recessive disorders. Recessive disorders which decreases the possibility of manifestation even in individuals with the presence of a defective gene. Sickle cell anemia affects what? What is affected in sickle cell anemia? Hemoglobin is affected. And the shape of the RBC is affected. RBCs are made up of what? Heme and globin. RBCs are made up of heme and globin. What is the function of RBC? What is the function of RBC? Function of RBC is to transport oxygen and carbon dioxide, predominantly for oxygen, because oxygen transportation happens mostly because of hemoglobin bound transport. Carbon dioxide transportation happens as bicarbonate, but that also in the RBC only, inside the RBC only, not in the plasma. Dissolved bicarbonates in the plasma is a minor mode of transportation of carbon dioxide. Now, one challenge that RBCs have to overcome is passing through the narrow capillaries. There are a lot of narrow capillaries. Big vessels, no problem, there is enough space for RBCs to flow. But When RBCs go into the splenic capillaries, the splenic capillary network, that is the capillary network in the spleen, is very narrow, which requires the RBCs to be flexible. The shape of the RBC, what is the shape of RBC? Biconcave. The biconcave shape allows the RBCs to squeeze through the capillaries. It allows RBCs to squeeze through the capillaries. So that requires RBC to have hemoglobin in a particular configuration, which gets packaged into the cell, giving the RBC its flexibility. Sickle cell anemia is a disorder where there is a substitution. It is a substitution problem. What is substituted? Valine by glutamic acid, no? Substitution of glutamic acid by valine in the 6th position of the beta chain of the globin, hemoglobin molecule, in beta chain of the globin molecule. So glutamic acid is replaced by valine. That changes the shape of hemoglobin. So when that changed shape hemoglobin is packaged into the RBC, RBC loses its biconcave shape and becomes sickle shaped. Now this sickle shaped RBC is not as flexible and pliable as the regular biconcave shaped RBC. Splenic capillaries will be difficult to pass for old RBCs. So when old and weak RBCs pass through the splenic capillaries, they will rupture. And that is the mechanism of ending the life of weak RBCs. So that you don't have to have weak and inefficient RBCs circulating in the blood. What will happen to the dead RBCs? How are they removed from the body? Heme is, iron from the heme is used up, is reused. Globin is broken down further to reuse the amino acids. The leftover RBC and all the metabolites will be converted into, in the process will be converted into bilirubin, and bilirubin will be excreted via bile. That is the fate. In sickle cell anemia, the sickled RBCs, even though they are young, will rupture when they pass through splenic capillaries. RBC production is going on normally. RBC production is happening normally, but because of the inability of the sickle-shaped RBCs to sustain the passage through splenic capillaries, they will rupture, leading to the reduction in the RBC count. When the RBC count is reduced, what do you call that condition? You call it anemia. Anemia is low RBC count or low hemoglobin concentration in the blood. If hemoglobin content is low or if RBC is lesser, that is anemia. Anemia functionally is the inefficiency of the blood to do transportation of oxygen to all parts of the body. Reduced oxygen carrying capacity of the blood is anemia functionally. At the cellular level, molecular basis is reduced hemoglobin count or the reduced RBC count. Reduced hemoglobin level or reduced RBC count is structurally anemia. So RBC count might be normal, hemoglobin content may be normal, but a person can be anemic. How? If with normal RBC count, with normal hemoglobin content, can a person be anemic? Meaning, can a person have the symptoms and the manifestation of anemia? What is the symptom of, what are the symptoms of anemia? RBC is supposed to transport oxygen. And when oxygen is available in adequate concentration, the body will function normally. If RBC count is low, the number of RBCs available are less. So even the hemoglobin in every RBC is normal. The total oxygen that the blood can carry will be lesser. Or if hemoglobin content is low, RBC count is normal. Is that also possible? RBC count is normal, about 5 million per deciliter of blood. But hemoglobin is less. Because of whatever reason, iron deficiency might be the most common cause. So every RBC is having less hemoglobin. That also will lead to decreased oxygen carrying capacity of blood. What can be the symptom? What will an anemic person experience? Fatigue. General malaise, you are not feeling energetic, you feel low, tired, you cannot do physical activity, you will not have focus and concentration. Brain also requires a lot of oxygen when it is active. So reduced concentration, all of that will be the problem. Can a person experience those symptoms without being low in terms of RBC count or hemoglobin concentration? Is that possible? If it is possible, tell me a condition where that might be possible. A condition which you have studied. If the person has carboxyhemoglobin, if a person has methemoglobinemia, methemoglobin, nitrogen bound to nitrates bound to the hemoglobin, hemoglobin becomes unavailable for transporting oxygen. When you measure hemoglobin, the hemoglobin level is normal. RBC count normal, but a person is having symptoms of anemia. Possible, no? Methemoglobinemia. Methemoglobin is nitrated hemoglobin. Hemoglobin to which nitrates are bound. They will also not leave. Affinity of nitrates to hemoglobin is not as strong as that of carbon monoxide, but affinity of nitrates to hemoglobin is stronger than that of oxygen. It will bind to hemoglobin strongly, more strongly than what oxygen can bind. So when oxygen is available, the methemoglobin will not release the nitrate, because nitrate is holding on to hemoglobin more strongly than oxygen is trying to catch hold of hemoglobin. That's all. So it is demanding hemoglobin's attention. Oxygen cannot win over that race. That is functional anemia. Most of the people, my theory is, most of the people in metropolitan cities are functionally anemic, including me. I live in Bangalore. Cities like Bangalore, Delhi, Mumbai, Kolkata, Chennai, Hyderabad, these are all big cities with a lot of pollution. Lot of pollution, no matter how many catalytic converters you have in the exhausts of vehicles. The pollution is so much that it is impossible for you to remove all the hemoglobin binding pollutants from the air. So you breathe air which is loaded with nitrates, you breathe air which is loaded with some quantity, some amount of carbon monoxide. You are always in a state of functional anemia. So though cities seem to be providing advantage in terms of opportunities, the productivity is low in people living in cities. Productivity is high in people living in cleaner environments. Your concentration will be higher, your focus will be higher, physical strength is higher. In that sense, advantage. But cities give opportunities for resource accumulation, money. Now we will read sickle cell anemia. You get that? What is the type of mutation that happens here? What is the type of mutation? Point mutation. There are two types of mutation. What is mutation? Mutation is change of nucleotide bases in the genes. That's all, no? Nucleotide base sequence. Okay? The gene sequence is altered. That is mutation. How can it happen? You can replace something. Point mutation is a particular base is removed and some other base is added. Will this change the length of the nucleotide sequence in the gene? No. Point mutation does not alter the length of the codon sequence. It will not alter the length of the codon sequence in the gene. But it will only alter the nucleotide base in that point. Is it possible that mutation happens but no change in the gene product is seen? Is it possible that there is mutation, some nucleotide base is removed and some other nucleotide base is added? The sequence has been altered at that point mutation has happened, but in the final product of the gene, there is no change. It has the same sequence of amino acids and the same protein and it is as functional as what it was. Is it possible? Mutation does not lead to any change in the gene product? How?
If 3 codons changes.
Hmm?
If 3 codons changes.
If 3 codons change. Codon is a triplet. If 3 codons change. Okay. See, that is possible only when the change does not lead to a change of the amino acid represented by the altered codon. Correct? If AUG is there, AUG is methionine. GUG, I think is also methionine. Correct? AUG, what is this property of the codon called? Degeneracy. Degeneracy is a property of the triplet codon that the same amino acid is represented by more than one codon. So if the codon that has been altered is also coding for the same amino acid which was coded by the original codon, then the sequence will not be altered. So that means point mutation need not necessarily lead to abnormality or a change. Point mutation can lead to a change, but point mutation can lead to no change in the final output of the gene. Those possibilities are there. Insertion and deletion will lead to what kind of mutation? If you add some nucleotides or remove some nucleotides, that is frame shift mutation. That is the gene reading frame is altered. Codons will get changed continuously from the point of mutation, from the point where frame shift mutation has happened. That is addition of some nucleotides or removal of some nucleotides has happened. Beyond that point, the entire codon sequence will get altered. Because it is like 1, 2, 3, 4, 5, 6, 7, 8, 9. That is the sequence. So 1, 2, 3 is one codon, 4, 5, 6 is another codon. You insert, let's say, A between 2 and 3. Now this codon will be 1, 2A, 3, 4, 5, 6, 7, 8. Every codon gets altered. If you remove 2, then it becomes 1, 3, 4, 5, 6, 7 changes. Frame shift mutation will always alter the amino acid sequence. Point mutation may or may not alter the amino acid sequence. So sickle cell anemia, to understand that, you need to know what is happening at the molecular level, what is happening at the functional level, what are the symptoms that you experience, why is it sickling, why is it called sickle cell anemia, why is it anemia in the first place. So what is the molecular level alteration that is happening? Glutamic acid is replaced by valine in the beta chain of globin of the hemoglobin. In the beta chain of the globin of hemoglobin, that is altered. So one chain is altered. When the RBCs, sickled RBCs rupture, it will lead to the reduction in the RBC count, eventually leading to the inability of the blood to carry out oxygenation process or the transportation of oxygen as efficiently as it should do. So that is a state of anemia. Sickled RBCs getting destroyed, leading to symptoms of anemia, leading to manifestations of anemia, you call it sickle cell anemia. How do you treat it? How do you treat it? For a very long time, whenever the anemia becomes very high, anemia becomes very severe, blood transfusion will add on the normal RBCs into the blood directly. The lifespan of RBCs is how much? 90 to 120 days. About 90 to 120 days is the lifespan of RBC. That means 3 to 4 months. 3 to 4 months. You infuse a unit of normal RBCs, normal blood into the sickle cell anemia person. So that will give a mixture of both sickled RBC and normal RBCs. The normal RBCs are carrying out the normal function, sickled RBCs are eventually slowly getting broken down. The person can maintain good health for a period of time. Then again, blood transfusion, again blood transfusion. Not the whole blood, the packed cell transfusion. It is packed cell transfusion that is done in sickle cell anemia. There are now more possibilities emerging for the treatment of sickle cell anemia, where the stem cells in the bone marrow, RBC producing stem cells in the bone marrow are harvested. Genetic manipulation is done, the normal gene insertion and the modified or the stem cells with the normal gene are put back into the RBCs, stem cell therapy. The normal stem cells are put into the bone marrow of the affected person. So those normal stem cells will give rise to normal RBCs. Stem cell therapy is being explored for the treatment of sickle cell anemia. There are complications with stem cell therapy. One, the stem cell of the patient himself needs to be modified. That still has ethical and legal complications. Gene therapy can be done. Normally, normal stem cells from a compatible donor. Stem cells from a compatible donor can be transfused into the bone marrow of the patient. Now, if that survives, that stem cell survives, that will continue to produce normal RBCs. If it doesn't survive, to the extent of continue to do it. But there are possibilities of treating sickle cell anemia beyond just blood transfusion. Okay? We will read it now, only one paragraph. Sickle cell anemia is an autosome-linked recessive trait that can be transmitted from parents to the offspring when both the partners are carriers for the gene or heterozygotes. The disease is controlled by a single pair of allele, HBA and HBS. Out of the three possible genotypes, only homozygous individuals for HBS, sickle cell hemoglobin, show the diseased phenotype. Heterozygous individuals appear apparently unaffected, but they are carrier of the disease, as there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. The defect is caused by the substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to the single base substitution at the 6th codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. Okay, is that clear? Sickle cell anemia? Okay, next topic. See, identifying what pattern of inheritance is seen in a given case is a favorite question from this chapter. If they give a case, some explanation, some inheritance pattern, and ask you to identify what type of inheritance can be seen there. Okay. Skip generation inheritance is seen in. Skip generation inheritance is a common feature of skip generation inheritance is a common feature of autosomal recessive. Skip generation and crisscross inheritance. What is crisscross inheritance? Crisscross. If a female is affected, the next generation male will be affected. If a male is affected, the probability is that the next generation female can be affected. Crisscross inheritance, skip generation inheritance, these are characteristics of autosomal disorders or sex-linked disorders. Whatever you guess, you have a 50% chance of getting it right, which is a very high probability. Sex-linked disorders. If it is an X-linked recessive character, affected mother will always give that disease to all her sons. Correct? So you have autosomal recessive, autosomal dominant disorders, X-linked recessive, X-linked dominant disorders. and Y-linked disorders or Y-linked characters. Y-linked characters are always passed on from fathers to sons, father to son. Can you name one such thing?
Hypertrichosis.
Hypertrichosis. What is hypertrichosis? Hairy pinna. You have seen some men having hairs on their pinna. Right? You've seen that? If somebody has it, 100% their son will also have it. So if any of your boys, if any of your fathers have it, you will also get it. But it's hair, no problem. If you feel it is cosmetically abnormal, you don't like it, remove it. Correct, no? Easy to handle. Then the texture of the hair. Texture of the hair is autosomal. Skin color is, skin pigmentation is autosomal or X-linked? Skin pigmentation, autosomal. What is special about skin pigmentation inheritance? It is a deviation from Mendelian inheritance. How? It is polygenic inheritance. It is polygenic inheritance. Why is it a deviation from Mendelian inheritance? Even if it is polygenic, dominant character will express, recessive character will not express. So what is the deviation? Mendel did not talk about polygenic inheritance. What is the deviation of inheritance of skin pigmentation or skin color or the height, both of which are polygenic inheritance, from Mendelian pattern of inheritance? Codominance. Where do you see codominance in height or skin color? It is a deviation. It is in contradiction to Mendel's law of dominance. Now you tell me.
So it is quantitative inheritance.
Skin pigmentation and height is quantitative inheritance. Mendelian inheritance, law of dominance is qualitative. What does that mean? What is qualitative inheritance, quantitative inheritance? See, Mendel was lucky because he did not come across any of these things in his experiments. He found tall plants or dwarf plants. He did not find intermediate height plants. If he had found intermediate height plants with the level of understanding at that point of time, he wouldn't have been able to do it. He wouldn't have been able to come to conclusion. So sometimes ignorance is bliss. Alright? Not knowing something will help you become capable of doing that something. Alright? The strategy I have given you for handling physics and chemistry numericals. Not doing it right now for the next 2 months is what will help you do it later. If you try to do it now, you will not be able to work on the capabilities of developing the skills to do numericals. See, quantitative inheritance is dependent on the presence of dominant alleles by numbers. If one dominant allele is present, 1 unit of character is expressed, let us say. If 2 dominant alleles are present, 2 units of character is expressed. You have that fair and lovely shade card. Shade card, the most useless brand. I'm telling it on camera. On record. Sue me, fair and lovely. Okay, anyways, there is a shade card. Different shades of the skin color. There are 3 genes for skin color. So how many alleles are possible? 6 alleles are possible. The color of skin changes with 1 allele, 2 allele, 3 alleles, 4 alleles, 5 alleles and 6 alleles. And 0 alleles also. If all the 6 are recessive alleles for skin pigmentation, then the person is an albino. If all the 6 are dominant alleles, then the person is negroid. It is not racial, that's how we are talking about it. Alright, this is not a racial slur. Anyways, fortunately for us, we are not so sensitive to racial comments. Otherwise, you call somebody tall, or you're abusing them. You should not do that, but anyways, we have not gone so crazy yet to be so sensitive and so self-centered. Alright, so from albino to negroid, there is a range of gradients of skin color. Correct? Somebody might be dark brown, wheatish, fair, very fair, and albino. All that is possible. This is quantitative inheritance. Mendelian inheritance is qualitative. Mendelian inheritance is qualitative. That means presence of a dominant allele is enough. Whether it is one dominant allele, two dominant alleles, doesn't matter. If a dominant allele is present, the expression of the character will be to the fullest extent. That is why in Mendelian characters, whether it is a homozygous tall plant or a heterozygous tall plant, both of them are equally tall. But if it was quantitative inheritance, you wouldn't have find two phenotypes, you would have find three phenotypes. No dominant allele for height, dwarf plant. One dominant allele for height, intermediate height plant. Two dominant alleles for height, tall plants. That would have been the case if it was quantitative, but because it was qualitative, one dominant allele or two dominant alleles, doesn't matter, the plant will be equally tall. So two characteristics in us, three things in us that is in contradiction to Mendel, and which are quantitative. One is two, skin color and height. Codominance and partial dominance, those are also against Mendelian principles. We see codominance in our blood grouping pattern. Blood groups. Blood groups is an example of codominance. What is pleiotropy? Pleiotropy. Just extension of it, some small things like that. One character being controlled by multiple genes is polygenic inheritance. One gene controlling more than one character is pleiotropy. Okay. So in that there is a primary influence and secondary influences. Primary influence is the main character that the gene will influence. Secondary characters are minor influences on those. Okay. Like I have promised, 3-hour sessions will have a 10 minutes break. Without extending 3 hours to 3 hours 10 minutes. So this is slightly over half time. The break should have been given 10 minutes ago. So we will have 10 minutes break. Now it is 10:50. We will break for 10 minutes. Come back by 11 o'clock. What is the next topic you want to discuss? Break for 10 minutes. Right? Let us assume the character to be something. Alright, let us assume the character to be something. Let us assume this is X-linked dominant. A male is affected. Assume it is X-linked dominant. If it is X-linked dominant, daughter is affected. Possible. Sons cannot be affected. Sons cannot be affected because son does not get the character from the father. Son does not get the X chromosome from the father. Son gets the X chromosome from the mother. So, what is the assumption that we started with? What if this was X-linked dominant? What if this was X-linked dominant? Is this possible? If it was X-linked dominant? Possible, why not? This fellow has X chromosome. So he might have got that X chromosome with a defective dominant gene. So he might express the disease. Who will he give his X chromosome to? The man gives X chromosomes to his daughters. This couple have 3 children. One is the daughter. So daughter is getting the disease. Possible in this case. You look at father and daughter, it supports this to be X-linked dominant. It can be X-linked dominant in that case, when you look at the daughter. See, in this generation, anybody else is affected? One son is also affected. son has only one X chromosome and he got that X chromosome from the mother. If this was X-linked dominant character, this fellow has got that X-linked dominant character from the mother. What does that mean? The mother also should be affected. Is she affected? She is not affected. So, not possible to be X-linked dominant. Rule this out. We rule it out. Pedigree analysis is a game of ruling out possibilities. You have to rule out possibilities. To rule out possibilities, see there are things like, if it is an X-linked dominant character, all the daughters will be affected. If it is an X-linked dominant character, all the daughters will be affected. If one of the daughter is not affected and the father is affected, it cannot be X-linked dominant. If it is an X-linked recessive character, mother is affected. If a female is affected, all her sons should be affected if it is X-linked recessive. If a female is affected, but at least one of the sons is not affected, then it cannot be X-linked recessive. So we have a case here where a female is affected. They have a daughter and a son. Son is not affected. So this character cannot be X-linked recessive. Not possible for X-linked recessive. So this is definitely not X-linked inheritance. So it has to be autosomal. Now, when we say it is autosomal, there are two possibilities. Either autosomal dominant or autosomal recessive. Autosomal dominant or autosomal recessive. Let us assume that it is autosomal recessive. Or let's say it is autosomal dominant. There are two possibilities. If it is autosomal dominant, the possibility is this fellow is either homozygous for it or heterozygous for it. If it is autosomal dominant. If he is homozygous for an autosomal dominant trait, then definitely all the children should be affected. Correct? Because one of the child is not affected. It is not autosomal dominant homozygous. If it is autosomal dominant, it has to be autosomal. If it is autosomal dominant, it has to be heterozygous for that. So this person should be either heterozygous for an autosomal dominant trait or homozygous for an autosomal recessive trait. Correct? This has to be either autosomal recessive or an autosomal dominant condition with the heterozygous condition. Assuming it is autosomal dominant. Will this lady have at least one autosomal dominant allele? Let us say it is autosomal dominant. Alright, assume. Capital A. Are you understanding? Trial and error combinations we have to see. Later I will tell you some tricks to remember, just to cross check. Homozygous is not possible. If it was capital A, if it was capital A, all the children would have got one of the capital A. Then they should have got the disease. Because one fellow has not got the disease, this fellow has small a. Correct? So this cannot be capital A. It has to be capital A small a or small a. These are the two possibilities. Now assume this is this one. This lady has definitely got one capital A because she is showing the disease. The other one is small a for sure because this lady is not showing the disease. Her mother is not showing the disease. So if it is autosomal dominant, she also should have shown the disease, which she is not showing. So this one is This is the combination. This fellow is small a. We are assuming it is autosomal dominant. Okay? We have ruled out X-linked inheritance. We are assuming it is autosomal dominant. So this fellow is not showing the disease. That means he does not have the dominant allele. So he is for sure this. This fellow is for sure capital A small a. Assuming that it is autosomal dominant. Now, look at the next generation. This couple have 2 children, one daughter and one son. Both of them don't have the disease. If it is autosomal dominant, then the genotype for both of them is small a, small a, small a, small a. If it is autosomal dominant, then these 2 fellows should have one capital A at least. Where did it come from? So can this be autosomal dominant? This cannot be autosomal dominant. It has to be autosomal recessive. Now cross check. We will cross check with whether autosomal recessive is possible. Now erase all this. When you are doing pedigree analysis, to do all your trial and error, you have to use pencil. Not a very dark pencil. Pencil which will give you marking without leaving impression. It should not give impression on the paper, but it should give you easily the mark, which can be erased also. So what type of pencil? HB, 2B, something. Maybe a 2B pencil will do. HB pencil will put a mark on the paper also. Now, we have logically deduced that this has to be autosomal recessive. Correct? Logically, we have come to a conclusion that this has to be autosomal recessive. We'll cross check. If it is autosomal recessive, what is the genotype of this fellow? 100% it is small a, small a. All the affected individuals we will write down. Small a, 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 small a. Correct? If it is autosomal recessive, all of them have small a, small a. This lady does not show the disease. So, she has to have at least one capital A. She has to have at least one capital A. She may have both capital A's. That is okay. But she at least has one capital A. At least one capital A. May have both capital A's. May have both dominant alleles. We don't know that. Can she have both dominant alleles? Why not? If she has both dominant alleles, how can she have 2 recessive alleles? One of the allele has come from the mother. So father has given the recessive allele. Because she is also showing the disease, another recessive allele came from the mother. So mother is a carrier. Mother is a carrier, father is affected. One daughter has got it. This person has not got it. So he might be either carrier or he has to be a carrier only. Because this lady is heterozygous, he is a carrier. This fellow is a carrier. This one is affected. Now, this one to be affected, their child to be affected, is there a possibility? There is a possibility if this one is also a carrier. Correct? Then both of them will give one small a. This is possible. This one is affected, this one is a carrier. Or this one might not be, this one has to be a carrier. This one has to be a carrier. Or he may not be a carrier also. He may be normal also. So this daughter is certainly carrier. This fellow also is a carrier. So this is possible. This problem is consistent. The logic is consistent with the disorder being autosomal recessive. Now with this analysis, we will look at some thumb rules. Number 1, if father is affected and at least one of the daughters is not affected, If father is affected and at least one of the daughters is not affected, it cannot be X-linked dominant character. It cannot be X-linked dominant because if father gives his X chromosome to all his daughters, if it is X-linked dominant, all the daughters would get that dominant allele from the father. So write down, father affected, daughter not affected, cannot be X-linked dominant. Cannot be X-linked dominant. Father affected, daughter not affected, cannot be X-linked dominant. We will add more of these rules with more problems. One more. Daughter affected, father not affected. Mother affected, son not affected. Mother affected, son not affected. Mother affected, son not affected. This case. Mother affected, son not affected. Cannot be what? Cannot be. If the mother is having the disease, mother is affected, but the son is not affected, then it cannot be X-linked recessive. It cannot be X-linked recessive because if it is X-linked recessive, then the mother is having two recessive alleles. Son will definitely get a recessive allele, and it is only one X chromosome that the male will have. Having one recessive allele for an X-linked recessive disorder will make the male get affected. If the mother is affected and the son is not affected, then it cannot be X-linked recessive. Daughter affected, father not affected. Daughter affected, father not affected. Again, it cannot be X-linked recessive. So that means, if there is an affected female, her father and all her sons will be affected if it is X-linked recessive. If either her father or at least one of her sons are not affected, then it cannot be X-linked recessive. For a female to be affected by an X-linked recessive disorder, she should have got one recessive X chromosome from the father. That would have made the father be affected. Understand? So X-linked recessive easy to identify. X-linked dominant easy to identify. So X-linked disorders easy. Autosomal disorders, autosomal recessive, do trial and error and cross check. Pedigree analysis, you should be able to get the answers easily. Let us keep it at this much now, the rules. Don't remember the rules. Even if you forget the rule, you should be able to apply logic. So I don't give you rules before you get the logic. I will give you rules only with respect to X-linked inheritance now. X-linked dominant, what is the condition? If the male is affected, all the daughters are affected. If the male is affected but at least one of the daughter is not affected, then it cannot be X-linked dominant. If a lady is affected, her father and all her sons should be affected if it is X-linked recessive. If father or at least one of the sons are not affected, it cannot be X-linked recessive. Now pick up another problem. There is one more here. Small pedigree. We'll see one more. You analyze and you tell me what can it be. Pedigree analysis is fun. Sometimes you get stuck up, you will not be able to conclude. Not always you can conclude with pedigree chart. You will need further analysis. Okay. This is just one family of 5 children. Normal son, affected son, normal daughter, normal son-in-law, affected daughter, is this specifically made for Meghalaya? Big families, 5-6 children and all. Okay, normal son, affected son, normal daughter, affected daughter, affected normal son. Okay, this is the chart. What can you rule out? You don't have to copy this. You can just look at it and analyze and rule out what you feel is not possible and come to conclusions. What can you rule out? It is definitely not any dominant trait. Autosomal or X-linked, forget. If somebody is affected and their parents are not affected, it is not dominant. Correct? It is not dominant for sure. So both two things are ruled out by default. Autosomal dominant ruled out, X-linked dominant ruled out. Then
X-linked recessive.
it is?
X-linked recessive.
Is it? Like autosomal recessive? Why do you say so?
Pedigree skip generation.
Don't complicate it. Look at the rules that I told you. Read the rules that I told you and apply it.
Daughter is affected, other is not affected, and it cannot be
See, whenever you look at a pedigree chart, look for affected females. Look for affected females. Then look at her father, and if she has, look at her children. She is affected, father and sons are not affected, then it cannot be X-linked recessive, ruled out. Children affected children, and look at their parents. Look at somebody who is affected and look at their parents. Somebody is affected but the parents are not affected. Then it cannot be dominant. So this is a 10-second problem for you. Children affected, parents not affected. It cannot be dominant. So by default, out of the 4, 2 is gone. 2 are gone. Autosomal dominant ruled out, X-linked dominant ruled out. Because parents are not affected. Write down that rule also. Children affected, child/children. So you should not get confused that all children are affected. Child/children affected, but both parents unaffected. But both parents unaffected, it cannot be dominant. In bracket, both autosomal as well as X-linked. Children affected but not parents, but neither of the parents are affected, it cannot be dominant, it has to be recessive. So two things you have to look at it when you see any pedigree chart. You will look at the affected child, look at their parents. Try to rule out dominant traits. Then look at the affected female, look at her father and sons. If either of them are not affected, done, over, autosomal recessive. So easy, no? Pedigree. Pick up any more, another, one more. Somewhere. You have something here in this? This one? What is this? It is fully colored. NCERT only has colored it. Still confused? You take a simple. Okay. Eliminate your confusion. We will take a small family. The husband and wife are staying together, joint family. It hit me so hard. It is only last week that I got to know about this view of looking at joint families. Somebody, I don't know who it is, but I saw it on some YouTube channel. He was talking about joint families. He was telling that in modern days, in the modern times, the joint family should not be looked at multi-generation families. The interviewer asked some podcast. Then what? In a modern urban family, it is a joint family if husband and wife are staying together. Husband stays in one place, wife stays in one place. Their working hours are different, so when husband is at home, wife is not at home. When wife is at home, husband is not at home. Somehow, once in 2 or 3 weeks, over weekends, they meet for dinner. That was somewhere outside, well, during transit. It's like airport meetings. Okay. Confusion. One daughter, 2 daughters, 1 son. Alright? Keep it this way. Now let us assume this person has an autosomal, X-linked dominant character. Or forget about dominant and recessive or anything. This person has XY chromosomes, this lady has XX chromosomes. Correct? XX chromosomes. Now, all the daughters will use color coding. The lady has blue chromosomes, the man has black chromosomes. Everybody is getting 1 X chromosome. All children will get 1 X chromosome from the mother by default. Daughters are getting 1 X chromosome from the father and son gets Y chromosome from the father. Correct? Is this clear? Understanding X-linked inheritance. Now, if you notice, son will never get X chromosome from father. Son will never get X chromosome from father unless it is a Klinefelter syndrome, XXY. Then there is a possibility of non-disjunction of XY chromosome and both XY chromosomes comes into one gamete, fertilized by fertilizing one egg and all that. That's normally a son does not get X chromosome from the father. Now, let us assume there is an abnormality in one of the X chromosomes. There is an abnormality in one of the X chromosomes which I will mention as a superscript, capital A. If it is autosomal, if it is dominant, if it is recessive, I will write it as small a. Let us assume one of this X chromosome has a dominant allele and the other one is a recessive allele. Correct? If the mother is affected. Now if this is a dominant character expressed, the presence of the dominant allele will make this lady get affected. So in this case, she would be affected. I will write a dot here. Easy to erase. Otherwise I have to erase the entire circle and then we will. Now this lady is affected. What is the probability of any of these fellows getting affected? All of them are getting X chromosome. They might get an X chromosome with an affected dominant allele or a recessive allele. So there is a possibility that these can be affected, cannot be affected. And because we are talking about a dominant character, if this X capital A is present in any of these, they will be affected. And if they are not affected, what does that mean? These fellows are not carrying the dominant allele. But we are not able to conclude. We will not be able to conclude in this case. Whether this is a dominant character or a recessive character. Right? Now reverse it. Let us assume this is normal, X chromosome on the lady is normal, but X chromosome on the father is affected. He has a dominant allele which gives rise to an abnormal character. It's a dominant trait. Now, by default, the X chromosome given by the X chromosome of the father goes to every daughter. So this X chromosome is definitely affected. This X chromosome is definitely affected. So this lady is affected. This lady is affected. This person is also affected. Correct? If it is X-linked dominant character, that dominant character gets passed on to the children, and whoever gets it will show the character, will show the trait. Yes or no? Now think about a situation where you are not seeing any disease in this father, but it is shown in the daughters. Neither of the parents, neither the father nor the mother has any disease, but you see disease in the parents, in the children. Not every children, one of the child is not affected, at least one of the, let's say only one is affected. Parents are not affected. Whatever is the character, whichever is the character that they got from the parents, they got one chromosome from father, one chromosome from mother. If it is a dominant character, then whoever is expressing it is having that dominant allele. They should have got it from their parents. They have got it from their parents. They are expressing the disease, but the parent is not expressing the disease. Means what? It is not dominant. If it was dominant, the parent also should have expressed it. Correct? The parent should have expressed it. Because the parent is not expressing it, and the children are expressing it, or at least one of the child is expressing it, it cannot be a dominant trait. Autosomal or X-linked, doesn't matter, but it is not a dominant trait. Did you understand? If it was dominant, it should have been shown in them. It should have shown up in them. Because it is not showing up in the parents and showing up in the children, the abnormal gene children have came from the parents, not expressing in them but expressing in the children. Though they have it, it is not being expressed. It cannot be if it was dominant. Because it is recessive, it is not expressing in them, and when it comes down to the children, both alleles become recessive, so they are expressing. Got it? So the first point of evaluation that you will do whenever you see a pedigree chart is, are children affected? See for their parents. Parents are not affected in whichever generation, doesn't matter. In any generation of the chart, you see children are affected, but both the parents are not affected. It cannot be dominant. Did you get the logic behind it? You understood the logic behind it? Now let us look at it another way. Okay. I will write normal X chromosome as capital X, abnormal X chromosome as small x. for the sake of simplicity. So that we don't have to do superscript and subscript and all. Daughter is affected. Now I don't have to use two colors. Daughter is affected. Okay. Let us use. She has. Let us assume it is an X-linked character. Daughter is affected. This daughter is not affected. Son is not affected. She got both. She has two X chromosomes. One X chromosome she got from mother. One X chromosome she got from father. From the father, what did he get? What should she get? She got the black small X chromosome. From the mother, she got. Mother had one normal X chromosome, one abnormal X chromosome. If it is X-linked recessive, she got one from the father, one from the mother. It is expressing. Now the daughter has the disease. Father is not having the disease. If it is X-linked recessive, she has got one of the recessive alleles from the father. In males, X-linked characters are expressed with one allele only. So if this small x was present in the father, he also should have shown the disease. But he is not showing the disease. Means what? Definitely not X-linked recessive. Father is showing the disease. No? Father is showing the disease. Look for her children. Look for her children. Right? Madam, it is pens we use all the time or we refill it? It's okay, this is working, no problem. It's working, this is working. There are many more things. So we will check for the children. Let's say they have 3 children, 2 sons and 1 daughter. One son is affected, the other son is not affected. One son is affected, the other son is not affected. Where do the sons get their X chromosome from? Who do they get it from? They get it from the mother. If it was X-linked recessive, mother is having both X chromosomes which are abnormal. So whichever X chromosome the son is getting, the son should show the disease. One of them is showing the disease, but the other one is normal. Her father has the disease. Okay, in that case, there is a possibility that it can be X-linked recessive. One of the son is showing disease. To this extent, okay, but one of them is not showing the disease. So it cannot be X-linked recessive. So if it is X-linked recessive and the lady is showing the disease, her father and all her sons should have the disease. Only then it can be X-linked recessive. If either her father or at least one of the sons don't have the disease, it cannot be X-linked recessive. So you have 2 very powerful tools to rule out 3 things. You look for affected people, look at their parents. If both of them are not affected, then dominant is ruled out. Look for an affected female, look for her father and sons. If at least one of them are, if even one of them are not affected, then X-linked recessive is ruled out. Alright. One of the parents is affected. There is only one son, one daughter. Son is affected. This lady is affected. No, you can't rule out dominant. You can't rule out X-linked recessive. You can't rule out X-linked recessive. You can't rule out autosomal recessive. You have to look for other clues. Alright. So you need to know the logic of navigating pedigree analysis questions. The tools will help you come to conclusion quickly. If you apply the tools, if it fits in, you come to conclusion very quickly, you answer it in 10-15 seconds, done, over, you save time, move forward. If you are able to rule out. If you are not able to rule out, then you will have to spend the time doing applying logic to exclude. eliminate the possibilities. Alright. Let us see if there is any good pedigree question. We can only create pedigree analysis questions, but it takes time, so we are lazy people. We will look for good pedigree analysis questions. You can, others also can look for it. Don't be so lazy to not look for it. Youngsters don't have the option to be lazy. Because the lazy elder people or older people want youngsters to work so that we continue to be lazy. Nobody should be lazy. See, anybody got any other questions? We'll do one more. One more. Look for a complicated pedigree analysis question. Got it? Study the given pedigree chart showing the inheritance of X3 trait controlled by G. What will be the genotypes of individuals A, B, C, D? See, what will you do when you have these kind of questions? See, this is it. Male, female, children. This is a very bad pedigree chart. You know why? This is a culturally wrong pedigree chart. Tell me why. Tell me why this logically nothing wrong, but we should not be using that pedigree chart in India. What is wrong? You don't know what is wrong. Give a write it and see. We will find out what is wrong. You got it? No? Give. I'll write it. This is keen in biology, physics wala. Alak Pandey, whatever.
Meeting between brothers and sisters.
They are showing marriage between siblings. Correct, no? See, I'm not making it up. Okay. So, we will correct it. We will use the chart, but we will not use the hopefully inadvertent error without any suggestive value. Okay. The question is, study the given pedigree chart showing the inheritance of an X-linked trait controlled by gene small r. What will be the genotypes of individuals A, B, C, D respectively? Who are A, B, C, D? This is A. This is B, this is C, this is D. It is an X-linked trait. The gene is small r. in such cases, don't look at the options. Don't bother to look at the options. We will write the genotype, the chromosomal composition of this. XY, XX. This is XY, XX, XY, XX, XY, XX, XY, XX. So it is small r, the affected gene is small r. This means what? Heterozygous condition. One is normal and one is affected. So it is normal X and XR. This is nothing, this is nothing, this is nothing. This one is XR. This is X and XR. Understanding it? This is X and XR. This is X and XR. So what is the genotype of ABCD? It's a simple straightforward question. What is the genotype of ABCD? A is XX. A is XX. See, even in that, in the options, two options have XX for A. The other two options have at least one XR. So those two options ruled out right away. Looking at the first one, B is XRY. So in one of the options, XRYR is given. The other one is XRY of the two remaining. Rule out the one with XRYR. What is left out is the right choice. Cross check with the other two being right. C is XRX, XXR or XRX. D is XY. That's all. So it is pretty straightforward and simple. So there are two more pedigree charts. We'll do one or two more. Is everybody clear with this? I want to get a chart which will confuse you between, which will not be able to come to conclusion. Let us see. Okay, this is good. Okay, we'll do one more. There are two charts given. In one question, two charts. It is like giving two questions in one question. This is chart A. This is chart B. Apply your thumb rules. See if you can rule out something. Oh, Alas Pandey, some problem. Look at this. In this also is doing the same thing. He is getting the last two children married to each other. You want to do scandal? Point it out and just do one kind of like crime story kind of report. How can you? This is B. Options are asking you, select the correct statement. A represents autosomal dominant, while B represents autosomal recessive. This is option A. Option B, A represents Y-linked trait, while B represents X-linked trait. Option C, A represents autosomal recessive, while B represents X-linked dominant. X-linked dominant. Option D, A represents autosomal recessive, while B represents autosomal dominant. Now, solve this. Go step by step. Try to eliminate possibilities, eliminate options. Two options are outright being done. Answer is right in the face. Three options can be easily eliminated. You don't even have to bother to cross-check whether the right choice is correct or not, because the wrong choices are so glaring on the face. Go back to the thumb rules. Okay, you take your time, build your logic. Come to conclusions, and whoever gets it will have to explain. I thought that was the incentive. Getting a chance to explain. But looking at the reactions, those who get it might not tell it. This is a 20-second question. Come on, you should not be taking so long. Is the right choice. Come, explain why. Why do you eliminate the others? Come. Correct. Option A is the right choice. Don't do all that. Tell me, why did you eliminate the other? No?
But this should not be violence. This should not be violence.
Because females are affected. Females are affected. So option B is ruled out. Strike it off. Strike it off. Strike it off. Take the pen. Great. It cannot be Y-linked because females are affected.
So this cannot be X-linked.
That cannot be X-linked, right?
Because generation is
Huh?
generation is
Hey, I did not talk about skip generation at all.
because I use that. Because I already know.
Now, how did you rule out option D?
Option B is autosomal recessive by the
No. You need to have simple logic to rule out. Option A is right, correct. Now read the rules I gave you. Read the rules I gave you and try to apply it. I have given you two very important rules. I told you how to navigate the pedigree chart. Got it? Come, tell me. Any opportunity to get on stage, you should get on stage and blast it off.
It cannot be recognized because mother is affected, but mother is affected or son unaffected.
Okay. See, what is the navigation rule I told you? Whenever you look at a pedigree chart, look at affected individuals and look for their parents. If the parents, both the parents of the affected individual are not affected, it cannot be dominant overall, neither X-linked nor autosomal. So option C and D. Option C is talking about chart B as X-linked dominant. Option D is talking about chart B as autosomal dominant. Both are not possible. Don't bother to cross-check with A at all, because certainly it is violating that rule. So C and D ruled out. Option B is talking about chart A being Y-linked inheritance. Not possible because females are affected. Option B ruled out. Then what the one left is option A. Option A is the one left out. Can it be autosomal dominant? It can be. Can B be autosomal recessive? Can be. Do we have to cross-check and verify in spell 10? No need, because we have eliminated the other three with strong evidence. That's all. Alright. So, pedigree charts can take more than necessary time. It can eat away more than the time required. Can you rule out? Chart B is definitely recessive. In this option, it is given as autosomal recessive. How can you rule out X-linked recessive from that chart B? Affected female and the previous and the next generation males, father and sons. Look at her. Daughter is affected. She is not yet having any son. Look at the father. Father is not affected. So it cannot be X-linked recessive. So in that sense, you confirm that autosomal recessive is correct. Can it be autosomal dominant? Yes, it can be autosomal dominant because one female is affected. She is heterozygous probably. Okay. If you are asked for the genotype of the affected females in chart A, will they be homozygous or heterozygous? Huh? Heterozygous. How can you be so sure? They are affected, but if they were homozygous, and we are talking about it to be dominant character, every child should have been affected, but you have few children getting affected, you are not getting affected. And dominant is what is given, so it has to be heterozygous, so that the one who gets the dominant allele gets affected, the one who gets the recessive allele is not affected. Clear? We'll end this, or do you want one more? We'll close this. Remember those two thumb rules. Number one, affected individual, both the parents. Individual affected, both parents not affected, dominant inheritance ruled out. Affected female, father and sons not affected, X-linked recessive ruled out. Or to confirm autosomal recessive by logic is difficult. So to conclude autosomal recessive, you should look for evidence to rule out dominant inheritance and X-linked inheritance. X-linked inheritance ruling out is easy. Alright? Because male has a hemizygous condition for X-linked characters. Is that clear? Which topic next? We have 20 minutes. Tell me one topic. Human Genome Project is just a mention. We don't go into the details of how it is done and all that. Skip that. Lac operon concept. Okay. Lac operon. Operon is a model of gene control of gene expression. How the gene expression is controlled. Lac operon is one set operon. Open it in the book. Wherever it is. Molecular basis of inheritance. Who has written this or compiled this? No author's name. Practice. Blood and sweat is here. Somebody sees this book, they'll feel, oh my God. You have put in so much effort. You have put in, the book has become old and digested. Yes. For the expression of gene products, for the expression of gene, there is a positive regulation and there is a negative regulation. Lac operon is a negative feedback regulation of the gene expression. We will just read it and try to understand the nuances of that. The elucidation of the lac operon was also a result of a close association between geneticist Francois Jacob and a biochemist Jacques Monod. Jacob and Monod is the model. They were the first to elucidate a transcriptionally regulated system. So, operons are the mechanisms for regulating transcription. Transcriptionally regulated system means it is a system of regulating transcription. So, when do we know the gene has to be transcribed? There is a stimulation, no? Like it's like the ovary is ready. The ovary has has all follicles with meiosis 1 complete, secondary oocytes being present there. When should the follicles actually start producing estrogen? The follicles can produce estrogen, but when should it start producing estrogen? It is a chemical clock. It is a chemical trigger. Something will go on for a while, making something prepare for the action of some other chemical, and that chemical acting on it will trigger a particular gene, goes on like that. Alright? So when that particular gene should be activated and when it should be stopped. That is the operon. In lac operon, here lac refers to lactose. A polycistronic structural gene is regulated by a common promoter and regulatory genes. What is a polycistronic gene? What is a cistron in the first place? Cistron.
Genes expressed.
A functional gene is a cistron. Correct? Cistron is a functional gene. What do you mean by a functional gene? That which has an observable expression. We know that it is producing something. That is a cistron. So, are we considering cistrons in the 5% of the DNA in our genome, which has some protein that it expresses? Cistron is a functional gene. Polycistronic gene means what? Polycistronic gene means there are multiple products being expressed by the activation of that particular gene. It is not a single product gene, it is a multiple product gene. In the case of lac operon, lac Z is one cistron, lac Y is one cistron, lac A is one cistron. So there are 3 cistrons. 3 cistrons producing 3 different proteins which will transform into 3 different enzymes required for the metabolism and assimilation of lactose. So cistron, operon, these are all terminologies. Such arrangement is and regulated by a common promoter and regulatory genes. Promoter, regulator and operator, there are 3 genes, promoter, regulator and operator segments. Promoter gene and operator gene are present. Promoter and regulator genes. Such arrangement is very common in bacteria and is referred to as an operon. To name few such examples, lac operon, TRP operon, ARA operon, His operon, Val operon, etc. The lac operon consists of one regulatory gene, the I gene. Here the term I does not refer to inducer, rather it is derived from the word inhibitor. Okay? And 3 structural genes ZYA. The I gene codes for the repressor of the lac operon. Repressor protein. So the regulator gene produces the repressor protein when it is activated. In the absence of lactose, the repressor gene is active. So it produces the regulator gene is active, it produces the I protein or the repressor protein, which will go and bind to the operator, which goes and binds to the operator. Operator is not available. Operator is the place which will allow for the transcription to begin. Alright? What is the what induces transcription? RNA polymerase. That is the site for the binding of RNA polymerase enzyme. RNA polymerase enzyme binding to the operator site will initiate the transcription process. From there it moves forward, transcribing the segment of the gene. We will read first. The I gene codes for the repressor of the lac operon. The Z gene codes for beta galactosidase, which is primarily responsible for the hydrolysis of the disaccharide lactose into its monomeric units, galactose and glucose. Y gene codes for permease, which increases permeability of the cell to beta galactosides. The A gene codes for a transacetylase. Hence, all the 3 gene products in lac operon are required for metabolism of lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. Lactose is the substrate for the enzyme beta galactosidase and it regulates switching on and off of the operon. Hence, it is termed as inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of permease. Remember, a very low level of expression of lac operon has to be present in the cell all the time, otherwise lactose cannot enter the cells. The lactose then induces the operon in the following manner. The repressor of the operon is synthesized all the time constitutively from the I gene. See, there are constitutive genes and inducible genes. There are inducible genes and repressible genes. Repressible genes are called constitutive genes. Constitutive genes are the maintenance genes. Genes which are active at all times unless they are repressed by some action. Inducible genes are inactive at all times unless they are induced to become active. Okay. So these genes, repressible genes are called repressible genes or constitutive genes are also referred to as dash genes. That's what I told you. If you have, what kind of work happens every day in your home? Does painting of the walls happen every day? Does changing the flooring happens every day? What happens every day? Cooking happens every day. Then cleaning happens every day. What is the one term that you will give to all the activities that happens in a home every day? Chores, okay. Then give me something else. This is a term used in institutions, hotels, restaurants, hospitality industry, and all that. For all the work, all the activities together. Yes. Huh? Management. Huh? Come on. Even 1 or 2 minutes to get this word is fine because you should not forget this word. What are these genes called? What do you call people who maintain all this cleanliness in restaurant, in hotels or institutions? Dash staff.
Working staff.
Correct. Housekeeping. So these constitutive genes are also referred to as what genes? Housekeeping genes. Alright? They are housekeeping genes, active all the time. You have to suppress them. If you don't want them to produce what they are producing, you have to suppress them. Otherwise, they are always producing what they are supposed to produce. Like the repressor protein. Regulatory gene is a housekeeping gene. It is continuously producing the repressor protein, which will keep the lac operon in a switch-off state. You have to turn it on. So this is an inducible operon. Inducible operon where you have to turn it on. Repressible operon is where it is working normally. Like the TRP operon is a continuously working operon. You have to stop it. You have to repress it. That is a repressible operon. Yes. This allows RNA polymerase access to the promoter and transcription proceeds. Essentially, enzymes of lac operon, regulation of lac operon can also be visualized as regulation of enzyme synthesis by its substrate. Remember, glucose or galactose cannot act as inducers for lac operon. Can you think for how long the lac operon would be expressed in the presence of lactose? How long? As long as lactose is present. Correct. Once the lactose has been metabolized, it will not be available for inducing. Regulation of lac operon by repressor is referred to as negative regulation. Okay. Lac operon is under control of positive regulation as well, but it is beyond the scope of discussion at this level. Okay. Then why did you mention? Just to tell that nothing is absolute. There is always flexibility. What you are assertive of today, you can be opposed to tomorrow. You don't have to be guilty of changing your stand or position. Okay? Today you have some clarity, you have a particular understanding of something. So you make your opinions and give your statements based on today's level of understanding. Tomorrow your understanding will change. You know something else. You get a different perspective. Just because you have spoken something today, do you have to stick on to that forever? You should be graceful enough to say, my perception has changed. I have a different opinion right now. So this is my current opinion. Will you stick to this forever? How can I say that? I am a human being, I keep growing. As I keep growing with more and more experiences, I will have different perceptions. For today, I have this perception, I will act according to that. Now you see how politicians operate. That's how real life is. It cannot be. See, politicians are a reflection of how you should operate in real life. Normal people are living in illusion. They fix up something and they will try to live to that. That is why they don't grow, because you have to drop what is not working for you, and you have to embrace what will propel you in life. Not necessarily with money, with anything. Because common people do not have the courage to drop what they hold dear to themselves, they are not able to move forward. To the extent that the people who don't move forward in life tend to hold on to their words also, hold on to their perceptions even though they realize it is wrong. Because I've told something some time ago, it's like promising to marry their daughter to friend's son. Daughter turns out to be a well-educated IPS officer. The one to whom daughter was promised when both of them were in their cradle turns out to be a complete wastrel. Will you still hold on to it? The father of that boy will come and say, "You had promised, my son has built dreams around it." Thank you very much. My daughter's life is more important than the promise I made you long ago. So we are not in a position to hold on to that promise, because this mismatch will not allow a happy coexistence. That's all. It is as simple as that. But this thing becomes plot for many movies. So, what is the operon for? Lac operon. Purpose. Produce enzymes responsible for metabolism of lactose. Where is the blueprint for this enzyme? On the gene. Enzyme should be produced, so gene has to be copied, that is transcription. The mRNA should be analyzed and transcribed into or translated into protein. So there is transcription followed by translation that needs to happen. Lac operon, when we are talking about it, we are talking about the conditions that will make transcription possible or not possible. That's all. So consider this as a factory. There is a production line. In this factory, there is a production line. There is a production line. This is the blueprint. This is the blueprint that has to be copied. Z, Y, A. This is where you will attach the copier. This is where you will attach the scanner. And this is called the operator region. This is called the operator region where the scanner is attached. Alright? And you have the promoter region and the regulator region. Regulator region. Now this regulator region is a housekeeping gene. This regulator is a housekeeping gene. This is producing a protein at all times. This protein is acting as a cover for this switch. Protein is acting as the cover for this switch. This protein is called inhibitor protein. Inhibitor protein produced by the regulator gene or the I gene. It is blocking the operator. If it is blocking the operator, the scanner cannot come and attach here. What is the scanner? Scanner is RNA polymerase. It cannot come and bind here. So if the scanner cannot come and attach here, it cannot copy this. What is the photocopy called? What is the photocopy in this case? The photocopy of this code or the recipe is mRNA. The scanner is RNA polymerase. The operator to which the scanner comes and binds is the switch. That is the switch. The switch is kept in the off position, or the switch is blocked by the cap. Cap is the inhibitor protein. Now, when lactose comes, when there is lactose in the medium, when lactose enters, what will the lactose do? Lactose will go and bind to the switch, bind to the plug. That is the inhibitor protein, and lactose will form a complex. So lactose will bind to this and remove this. What happens when lactose is there? The switch is exposed. The switch is exposed for the scanner to come and attach. But this fellow is a housekeeping fellow. He is continuously producing these proteins. But because there is lactose available, how many ever proteins are produced, lactose will bind to that and not allow it to go and bind to the switch. Lactose will not go and allow the plug to go and bind to the switch. The repressor protein or the inhibitor protein is binding to lactose, or lactose is binding to the inhibitor protein. So what is lactose now? Lactose is making it possible for the switch to get activated and for the scanner to come and get connected to the switch. So lactose is the inducer. INDUCER, inducer. It will induce the initiation of scanning and copying. That's all. How long will this switch remain on? How long will this switch remain on? As long as this plug is not coming and binding to this. How long will the plug be kept engaged?
As
long as there is lactose. How long will the lactose remain? For as long as these fellows need to break down lactose. Z will produce beta galactosidase which will break down lactose. So if lactose is continuously coming in, this will remain on, producing the enzymes which will break down lactose and make lactose available for other purposes. When lactose coming into the cell becomes lesser, the lactose available to block this repressor will become less. The enzyme that is already there which is breaking down lactose will continue to break down lactose. The protein, when it is free, will go and bind to operator segment. So this is switching off. You understand? So this is inducible operon, lac operon. Who induces the lac operon? Who is the inducer for lac operon? Who is the inducer? Lactose. Inducer is the lactose. That's all. This is all. That's the end. All of these concepts, this one, protein synthesis, all of these are good script for drama, stage drama. You can make this into a drama. Each of these can be characters. This one. All these fellows. Now you see, these fellows are not allowed to express. They are asking, boss, can we talk? No. Keep quiet. Operator is telling, because switch is off. Boss, can we talk? No. Keep quiet. Lactose comes. Lactose looks at them. These people are not being allowed to express. I need to do something. Lactose looks at it. Somebody is telling this fellow to stop them from expressing. So lactose goes, fights and catches hold of this repressor fellow and removes him from the switch. Now this fellow is not influencing the operator. These fellows will ask, boss, can you speak? Yes, go ahead. Now they start expressing. They start speaking, and what do they do now? They will come to lactose and start beating him up. And when lactose is finally down, the repressor goes and again binds to them. Now these fellows are again asking, boss, can you speak? No. Whom did we beat up? We beat up somebody. Who Who was in the first place responsible for our expression? You will never know who your true friends are. Moral of the story. Alright, okay. We will close for the day now. Tomorrow again, think of a few more topics. Tomorrow I will be leaving. So maybe I will conclude the session by around 12 o'clock. So be prepared for no 10-minute break. Today afternoon, I will get to speak to about 8 more students. 2 to 4, I'm here only. I'll be here till night. This is a good place for me to sit and work. So we'll complete that interaction also. Tomorrow we will discuss few more topics and done.